So now we are talking about osmosis and red blood cells, which are animal cells. For that experiment, we are taking 3 test tubes with differently concentrated solutions. Test tube A has a very dilute solution, test tube C has highly concentrated solution, and test tube B has a medium concentration. We are adding samples of red blood cells here, and when we look at a cellular scale, we see the red blood cells with the background of differently concentrated solutions. The very dilute solution can also be labeled as a hypotonic solution. The word hypotonic means low concentration. Isotonic means same concentrated solution, and the word hypertonic means very high concentration. Here in this context, we are trying to explain in terms of water potential. So the word hypotonic means a solution which has a very high water potential. So water potential outside, which means the water potential in the test tube is very high. In the isotonic one, the water potential outside, which means the water potential in the test tube is equal to the water potential inside. And in C, the water potential is going to be very low. Water potential outside is very low, which means there is very less water in the test tube compared to the red blood cells, obviously. In the hypotonic one, since there's a high water potential, the water is going to move inside the red blood cells because water always moves from high to low. In the isotonic one, the amount of water moving inside is cancelled by the amount of water moving outside. But in the hypertonic one, all the water from the red blood cells will move outside into the test tube because the test tube water potential is already low. So we can expect what's going to happen. In the hypotonic solution, more water moves inside. Inside what? Inside the red blood cells. So more water moves inside via osmosis. What happens? Cells swell up and finally burst. So red blood cells are going to become so much bigger and they're going to finally burst because of water coming in. In the isotonic one, cells remain the same. As the amount of water, you can say, which is moving inside is equal to the amount of water moving outside. So the rate of water moving inside is cancelled by the rate of water moving outside. What happens in the hypertonic one? In the hypertonic one, more water moves outside the RBCs. And ultimately what happens? Cells shrink down because they lose all the water from the cytoplasm, as you can see here. In the hypotonic one, cells swell up and they finally burst. This is what we call hemolysis, where the entire cellular contents just spread through the solution. In the isotonic one, cells remain the same size. While in the hypertonic one, since cells lose their water, they shrink and they become so much smaller. And this all happened because the red blood cells had no cell wall and they can't withstand the high pressure. They are not like plant cells which can handle turgor pressure. They don't have cell wall, so either they burst or they shrink depending on the water potential. I hope these ideas are clear to you guys. Stay tuned. Thanks.